Okay, hello everyone. So before we go on for chapter 5, let's go through the quiz participation assignment for chapter 4. Okay, so number one, the right answer is D. Okay, polymorphic locus doesn't have to be the same, identical in sequence, okay? And also doesn't have to give you a different phenotype, okay? May not have a phenotypic effect. So number two is interactome, okay? Independent protein complex. Okay? Then liver cell and brain cell should be same in genome, right? But all transcriptome proteome interactome are different. So among multicellular eukaryotes, species with a larger genome tend to have a larger content of repetitive gene, right? That is true. So mitochondria, the regulatory element, except it's called D loop or regulatory element. So we going through D loop in the replication section. Okay, sleep can be detected with GWAS. So mitochondria, chloroplast, inherited maternally. So difference, genome is complete set of genes, right? Everything there. Okay, but the transcriptome is expressed ones, collection of expressed gene, right? And also transcriptome is cell type specific, okay? So when you answer the question, before answer that, read the question really carefully. There are two questions here, okay? So this is about synteny. So same or similar gene in the same order in two different chromosomes, in the chromosomes of two different species. So mitochondria and chloroplast are believed to come from endosymbiotic correlations with the host. Okay. Now, so let's go for chapter 5. So genome sequences and evolution. Genome sequences and evolution. So a lot of organisms' genome have been sequenced, and we know the number of genes in many, but not a lot. But I mean, not every, but still, we've sequenced many organisms so far. So there are 500 genes found in parasitic bacteria. Okay, what's the parasitic bacteria? It's parasite. What's the definition of a parasite? A microbe or organism that completely relying on host to propagate, right? Parasite cannot live by themselves. So parasitic bacteria is bacteria that only live inside the host cell. So on the other hand, free living bacteria, they can live outside, okay? They can propagate freely. So their genome contains 1,500 genes. And this is a single-celled eukaryote. This is a fissionist. So about 5,000 genes are found in the genome. And this is a nematode, multicellular, still simple eukaryote. 13,000 genes, and plant 25,000, human or mammals around 25,000, okay? So this is number of genes are relative or average number, okay? Each species, there are different number of genes are in the genomes. So here's the representative species and the genome size and number of genes, okay? Let's cut right after the E. coli, okay? This is a line, all upper is prokaryote, bottom is eukaryote. Okay, let's look at prokaryote. Mycoplasma, this is a parasitic bacteria, okay? So their genome is 0.58 megabase, number of genes is close to 500. As you can see, genome size become larger and the number of genes become more, okay? In prokaryote, you see more genes in more larger genome size, right? But in eukaryote, it's not the case. We cover this later, okay? And again, for the living bacteria, so have about 1,500 genes, but parasitic bacteria is 500 genes. So it's believed that 500 genes may be the minimum number of genes for a living organism. Okay, just just hypothesis. Okay, in prokaryote, the genome size is correlate with the number of genes. Okay, in prokaryote. Okay, so purple is obligate parasitic bacteria, red is other bacteria, and the green scale is archaea. See the spot, right? It's very well correlated in prokaryote. Another characteristic of a prokaryote is the existence of pathogenicity islands. Pathogen, what's pathogen? Pathogen refers to microbe that can infect human or host to cause disease. Okay, this pathogenicity is in some bacteria characterized by the existence of pathogenic island. This is segmental DNA, contains several genes that are related to the pathogenicity. Okay, so those genes play a key role for a certain bacteria to infect the hosts. Okay, if you don't have it, very likely they are not pathogen. Okay, so this pathogenic island exists in Salmonella pathogen. This is example, Salmonella pathogenicity island 19. Close species, Salmonella enteritidis, this species don't have this area. Okay, this pathogenicity island can jump from one bacteria to another, okay, by the mode of horizontal gene transfer, horizontal transfer. Okay, so horizontal transfer means a DNA from one bacteria to another bacteria by bacterial conjugation. Okay, we're going to cover the conjugation in detail later. Okay, so in eukaryote, the genome can accumulate the genetic variation through sexual development or meiosis, but bacteria don't have the equivalent things to meiosis. Instead, a gene directly can transfer from one species to the other. This is, let's say, this is a laboratory E. coli strain. You use E. coli to propagate the plasmid, right? Plasmid has a selection marker, antibiotic resistance gene, right? And you dump this live E. coli through sewage water. There, in nature, and pathogenic Cryptococcus pneumoniae by chance uptake the antibiotic resistance gene from the experimental E. coli. Then this bacteria become antibiotic resistance. Then if this bacteria infect a certain human, then antibiotic doesn't work. This is the consequence used in the laboratory in the strains that are resistant to antibiotics. Okay, there's a clear and strict rule: you can't dump laboratory strain through sewage water. Okay, otherwise, chaotic thing could happen. So in eukaryote, okay, so several relative eukaryote species has been sequenced and determine the number of genes. So in yeast, 6,000 genes. In nematode, 21,000. Fly, 17,000. Arabidopsis plant, 25,000. It's like random number, right? Look at this. This is genome size. Human is here. Mouse is here. Y-axis is number of genes. Human genome is way, way larger than those of rice and Arabidopsis. Okay, We believe we are much more complex organism. This is Arabidopsis and this is rice. But look at this, the number of genes. Rice, even smaller genome, has more genes than human do. So as I can tell, this is genome paradox or C-value paradox. In eukaryotes, okay, genome size does not correlate with the number of genes. Okay, again, in eukaryote, genome size does not correlate to the number of genes. Okay, C-value paradox. What's C-value? C-value means picograms of DNA in haploid nucleus or half picograms of somatic cell. Somatic cell believed to be diploid, right? So that's actually the same number. So C-value paradox exists in eukaryotic genome. So people compare the two the genomes or its average genes in two different model yeast species. Saccharomyces cerevisiae and Schizosaccharomyces pombe. Let's see. Saccharomyces cerevisiae is baker's yeast or budding yeast. You see, those cells are budding out, right? This is how they grow. And Schizosaccharomyces pombe, this is a fission yeast. They divide by fission. Okay, they're clearly different, but both the single-celled eukaryote organism. The average gene look like this. Okay, so 5% of baker's yeast gene have 1 intron. It's only 5%, but here 43% of S. pombe gene have 2 intron, average 2 introns. It's a big difference, right? And the average gene size is like 26 base pair different, but intergenic space is very different, right? 2.5 times larger in S. pombe compared to that of S. cerevisiae. Okay. So as you can see, the average gene size or architecture of genomes are different. They vary from species to species. So gene can be expressed monocistronic mRNA or polycistronic mRNA. What is this? So monocistronic mRNA means one messenger RNA encodes one polypeptide or one RNA. That's it. So most eukaryotic genes eukaryotic messenger RNAs are monocistronic. I say most, not all. On the other hand, polycistronic means one messenger RNAs can be translated into multiple different proteins or can become multiple different RNA. Look at this. This is one messenger RNA, okay? One messenger RNA, regulatory gene, a promoter area. So this is expressed, this messenger RNA will give you protein A, B, C, and D for different proteins. So more than one gene are expressed from one messenger RNA. It's polycistronic. So normally this is in case of prokaryotic, okay? But there are exceptions. So one ribosomal RNAs in eukaryote expressed as a polycistronic messenger RNA, okay? But in general, in eukaryote, monocistronic. In general, prokaryote, polycistronic. So genes can be categorized by their functions. Enzymatic activity here, nucleic acid binding activity, protein binding activity, transcription regulation, ion binding. So I'm not going through everything, okay? That just you can categorize their activity, okay? But still, a quarter of gene function are not known. Okay, this is a function of Drosophila gene. Okay, Drosophila also is a model organism, right? Very simple insect model, but still, we still don't know the quarter of the gene's function. But in overall, you can see what kind of genes are there, right? And when you compare the entire genes of the genes of multiple different eukaryotic system, okay, you can find about 20% genes are all common in common in all eukaryotes. Okay, this is the Drosophila gene, fruit fly genome compared to other eukaryotes. 20% you can find all eukaryote cell. Okay, so those called orthologs, right? Related gene in different species. Okay, they function same way. That is ortholog. Okay, and additionally, there's additional genes in multicellular eukaryote. Okay, so single-celled eukaryote, again, yeast, they are single-celled eukaryote. But in multicellular eukaryote have additional genes, and the others, like 70% are species-specific, genus-specific genes. Okay. Human genome. What do we have in the genome, right? So more than 50% human genome is reparative DNA. They are non-coding. Okay, there's no gene there. See, exon plus intron is the gene, right? Only 25%, only quarter of human genes define human genomes. Genome defines gene. So they are the intergenic area and reparative DNA. Even coding area is on exon is only 1%. Okay, so human genome contains largely, really large non-coding area. It's very interesting, reparative DNA. What's reparative DNA? Okay, those are trace of ancestral invasion by virus genome. Okay, so we have viral DNAs in our genome, foreign DNA. Okay, it's very interesting. So this is average human genome, human gene architecture. Okay, so you don't have to memorize this, just look at this. So average human genes have 9 exons interrupted by 8 introns. Okay, this is average exon size. Okay, average 3' UTR size, average 5' UTR size, and average 1 intron size. So previously, we believe human genomes encode 50 to 60,000 genes because human proteome has 50 to 60 proteins, different proteins, right? One gene, one protein. But that's not true. So after sequence then analysis, then if people found human genome technically encode lesser genes originally expected. So why we have more proteins than the number of genes? So about 60% of human genes are alternatively spliced. Alternative splicing can give you the different, functionally different protein, right? So among these, 80% of them actually give you the different protein sequence. Okay. So again, so here's this is reparative gene. 45% is transposons. Okay, transposons are transposable element. This is moderately reparative and large duplication and simple repeat. Those are all virus-like sequence. Okay, and also human genome contains some duplications, gene duplication. And the Y chromosome. In animals, Y chromosome has its own characteristic. Okay, Y chromosome is really small compared to the size of X chromosome. Okay, Y chromosome only has about 60 genes. This is about the human. Okay, and genes are present in multi-copy. See, multi-copy genes. There are a lot of genes. It's not a lot of genes. So single gene exists in a multi-copy. Why this is the case? So gene in Y chromosome play key role to provide male characteristic. Okay. If there's a single gene provide that function, let's say if you have a mutation, then it's done. You completely lose the male characteristic. But having multi-copy, okay, so active gene still can be maintained. during long time of evolution, right? So you can accumulate some mutation here and here, but you still have active non-mutational genes existing, right? So this is the general architecture of Y chromosome. Okay? There's X chromosome transposed area, okay, the degenerative X chromosome area and centromere, and the ampliconic region. This is Y chromosome specific region. Okay. So one of the key gene in Y chromosome to confer male characteristic is SRY, sex determining reason Y, or testis determining factor, TDF. This is a transcription factor. Okay. How do you know this is a key transcription factor for male? Okay. So to know the function of a gene, what is the best way? To delete the gene to see what's defect. That way you know the function of the gene. So you can do this in human. And we use model mammalian species, mouse. So scientists knock out this transcription factor in male gene. So this male, genetically male, but doesn't have SRY gene, knocked out, okay? This male genital area looks like that of a female. Okay, this is female, real female. This is SRY knockout. So very similar. And in addition, this knockout animal exhibit estrous cycle and also behaves shows a female copulatory behavior. Okay, just don't have the SRY transcription vector, but seems like he believes she's a female. People did other things. Okay, now this is a wild type normal male, XY male. Okay. Now this is originally female, but scientists not in SRY genes there. Okay. So now this female express SRY transcription factors. So now you can see this is this female XX male show male genital organs. Okay, so single transcription factors really change their morphology, right? Really did in the governs the male morphology. Okay, but they are not fully functional as female or fully functional as male. But this is a substantial function, right? So some genes are essential. As I mentioned, SRY gene was knocked out. That means that gene is not essential, right? But some are essential. How do you know some genes are essential? That means you can't generate knockout. That means the gene is essential, okay? Then is there a specific function related to be, for a gene to be essential? So look at this. People categorize the gene and found the essential genes there. Transcription factors, the red bar is essential genes. All transcription factors, some are essential, some are not. Protein synthesis, proteins, some are essential, some are not. If cell communication, some are essential, some are not. So among all group, like essential genes are just distributed randomly, right? It's not like certain functions are related to the essentiality, right? Essential genes can just selected randomly. It's very interesting. And also, some genes can knock out, but about half of them don't give you the specific phenotype. Genes there, you knock out, there's no phenotype. So it's possible that one, two or more genes can be existing there, redundant gene, right? There's multiple genes. And still, we don't understand why we, can knock out the gene, right? So we said mutation is detrimental, point mutation causing mutant phenotype. But see, in Drosophila, about 90% gene, so there's no detectable effect if you knock out that. See, it's possible we just didn't find the appropriate condition to see the phenotype. But still, so this is very interesting. So we still don't know why we don't have phenotype. Okay. So some genes can be knocked out, of course, right? But cannot be combined. This is a synthetic lethality. Because there's a gene A and B. Okay, you can knock out gene A, still alive. You can knock out gene B, still alive. But if you knock out both genes now, it's lethal. So this is a synthetic lethality. So people do the synthetic genetic array analysis. Just find which gene combination give you the synthetic lethal phenotype. Okay. So people use Buddingist because it's a model organism. Then why we do that? Why you care about this, synthetic lethal genetic arrays? Because this can be applied to find the treatment for cancer. So cancer is a genetic disease, right? If you have a certain malfunction in the malfunction in certain gene, they may come out as a cancer, right? In normal cell, there's gene A, B. We know it is a synthetic lethality relationship. Okay, so there's a drug can inhibit B pathway. But without B pathway, normal gene can still propagate. It's fine. But let's see. If there's a cancer cell caused by malfunction of A pathway, okay, this is still alive. A pathway is malfunction, but still B pathway is there. But if you inhibit B pathway on top of A by using a certain drug, now your treatment result in synthetic lethality. Okay, this cancer cell may die, but these drugs don't kill normal cell. It only targets cancer cell. Okay, so again, cancer cell caused by this aging malfunction, but by targeting the different gene, then you can find the treatment for the specific cancer, right? That's why we care about synthetic lethality. So genes are expressed evenly. All genes are expressed at the same time, and all genes are expressed at the same level? No. Okay, they are differentially expressed in different genes. Okay, in the same gene, some genes are expressed really a lot, some are not. Okay, people measure the level of expression by using the hybridization technique between messenger RNA and cDNA. Okay, so if you have a more messenger RNA, that means you have a quicker hybridization. So quicker hybridization means you have a more RNAs there, and slow hybridization means you have a less RNAs there. Okay, this is RNAs extracted from chicken oviduct cell. Then they found albumin RNA hybridized really quickly. Okay, but some are hybridized in the midway, some are really late hybridized slowly. Okay, about 13,000 is most messenger RNAs. I said most messenger RNA hybridized really slowly. What does that mean? Most genes are expressed at very low level, right? And some genes hybridize in the midway. So this is about 7 to 8 messenger RNA. So only several genes are expressed moderately. Then one gene, in this case, albumin gene is expressed really high. Okay, so these lowly expressed messenger RNAs, scarce or complex messenger RNA. This actually 1 or 2 copy. That's it. 1 or 2 messenger RNA. That's it. Those are complex because why? Most genes are there, right? There's really complex messenger RNA. On the other hand, abundance of this RNA is large more than this, right? And abundance of albumin is more than this. So the abundant messenger RNA is a specifically high amount of messenger RNA in a certain cell, or in the same cell or certain condition. Okay. So this is called, and housekeeping genes. Technically, theoretically, housekeeping gene express every single genes evenly. Okay. So those genes are used for expression analysis control. When you measure your gene of interest, you should measure in relative to housekeeping gene, okay? Otherwise, it's a relative term. Gene expression is high, low, middle is a relative term, okay? So there's several ways to measure gene expression, right? Northern blotting, you measure the amount of messenger RNA. Western blotting, you measure the amount of protein. So RT-PCR, you also measure amount of messenger RNA. Those are the way to measure the expression. To do all of this, you should, use housekeeping gene as an expression control. Okay. So abundantly expressed messenger RNA, for example, chicken oviduct, albumin messenger RNA is abundantly expressed. That is called luxurigen. Okay. So know the difference between luxurigen, housekeeping gene, and scarce gene. Okay. So I provide the link for movie clip for housekeeping gene and luxurigen. Okay. You can find the information there. It's not long video clip. Okay. Again, so a lot of genes are common genes, but they are really, the level of expression is really low. Only luxurigen highly expressed. So DNA microarray, it comes again. So DNA microarray detect the level of expression at the same time. A lot of genes, expression at the same time. Okay. So in this case, one color expression. There's A, B, C, three different conditions. You have microarray tire. You collect the messenger from each condition or each cell, then dump this, then look, see the color intensity. Okay, you label the messenger with the green fluorescent molecule. If you have more green, that means gene is expressed high. You see the different intensity of color. Okay, so microarray also provide you the unmasked gene expression measurement. Okay, what's the benefit of a microarray? Here, I mentioned you can measure the gene expression by using northern blotting, western blotting, or RT-PCR. And microarray, right? So what's the benefit of microarray? You can do that multi-cell at the same time, multi-genes at the same time. So northern blotting, RT-PCR, and western blot, you only do one gene at a time. But here, you can look at this tire. You see a lot of genes, see. So you can save the time, of course. But this is, of course, expensive. But there's always an up and down. But if your project is to see the expression of a human genome in certain gene condition, if you do the northern blotting, you have to do 20,000 genes, right? But microarray tires, so you only purchase about 20 tires, you can do that. So how you can apply microarray to human disease? Okay, this is an example. So you can see the gene expression change in human condition. Okay, so this experiment is to look at the 12 genes expressions in 59 invasive breast tumor cancer women. Okay, so they grouped two. So one group is breastfed patients, the other group is not. Okay, so look at this upper line. Okay, upper red line is breastfed female line, blue line is non-breastfed female line, and they measure each individual gene's expression by using microarray, then rearrange that. So look at this. This is called heat map. Red means high express. Okay, more red means more express, and blue is less express. Okay, more blue is less, more, much less expressed. So just look at this heat map carefully. Can you see an imaginary vertical line to separate here? Okay, and also another imaginary horizontal line here. So there's a line here, here, right? And look at the upper. This is more breastfed female and more non-breastfed female, right? So it looks like breastfeeding may be correlated with the higher expression of these 9 genes, right? And lower expression of these 3 genes, right? So as you can see, of course, you may expect a really clean cut, but human individuals are genetically really vary. Okay, it's not, that kind of cutting is not gonna be existing at all. But see, this is really relatively clean. Okay, so clear correlation between breastfeeding versus the expression of these 9 genes. So DNA sequence evolved by having mutations. Some mutations are synonymous. Some are non-synonymous. Synonymous means same amino acid, okay? So for example, if we have a mutation in the 3rd DNA base, there's no amino acid, it's still leucine, right? That is synonymous mutations. Mutations that do not change amino acid, that is synonymous mutation. On the other hand, mutations that result in change in amino acid is called non-synonymous amino acid. Okay, syn means same, non-syn means not same amino acid. Okay, syn, same amino acid. Okay, so in evolution, synonymous mutations accumulate faster than non-synonymous. 10 times faster. Look at this graph. Okay. As time goes on, accumulation of mutation in synonymous is really shifted from non-synonymous. Okay, it's very, very different. So why? There's no amino acid sequence changes, right? So that's why this mutation accumulates faster than non-synonymous amino acid change, non-synonymous mutations. Okay. So by comparing this in a mutation ratio, people predict when this mutation occurs. Okay, so this is called molecular clock, because we know the ratio of a single mutation, time of the time required for the single mutation accumulation, right? So by using as a molecular clock, then you can move back when this mutation occurs. And also you can predict by comparing 2 sequences in 2 different species when evolutionarily they are diverse. Okay, let's look at the globin gene complex. Okay, so you remember like gamma globin, right? Globin in plant and exon fusion generate myoglobin gene. Okay, you should remember this from chapter 3. Okay, I'll show you that figure again at the end of the lecture, this lecture. And this gene, okay, undergo duplication, gene duplication, make another copy and diversification. So first duplicated, there are 2 genes. It could be used to be alpha alpha. Now second alpha become beta because it accumulates DNA sequence changes. So gene duplication and diversification, right? Now this 2 genes unlinked, separated. Now you have alpha and beta gene, okay? Then this gene clusters expanded. That means another duplication, alpha 1, alpha 2, and another duplication is another duplication, right? Now cluster, alpha cluster and beta cluster formation, okay? Look at this. You can observe this in plant, like in plant, okay, you can observe this gene in insect, okay, and you can observe this alpha beta linked gene in amphibians and fish, and you can observe these alpha clusters, beta clusters in mammals and birds. So you can predict how long it might take for this happen. Then you can generate this x-axis time clock. Okay. Now this may happen 700 million years ago, and from amphibian, ancestral reptile, ancestral mammal may come out about 250 million years ago, things like that. Okay. So this is how people generate evolutionary phylogeny. Okay. So this is so they constructed this phylogeny by using molecular clock, not like measuring the carbon trace measuring method, just by using molecular clock after analysis of DNA sequence. Okay. I'm not saying this is correct, but this is one way you can predict the time of evolution. So here, from common ancestor, reptile, birds, and mammal, reptile, bird as one group, and mammal come out about 300 million ago. So before that, they share the same common ancestor. And here, reptile and bird are separated about 200 million years ago, just based on the molecular clock after the DNA sequence analysis. Then you may, so we just meant, so talked about the synonymous, non-synonymous mutation, right? That is about coding area. How about intron? Where is the intron from? Okay, so there's two ways, intron late model versus intron early model. The intron late model is, that is hypothesis that intron exists there. Okay, and it just added to the son gene. Okay, so early gene, ancestor gene may not have any intron, but intron is now incorporated there. Second is intron is already there, just there. Okay, so interrupted gene here. So this is ancestral gene, and messenger come out. This is regular, and how this is supported, because in this intron, one exon can be correspondent to the one functional domain, right? And also different gene can have similar or same exon. So that is, called exon shuffling. Exons may shuffle around together with intron. Okay, so this supports the intron early hypothesis. Intron is already there and just move around together. Okay. So some genomes are large. As I mentioned, there's a C paradox. So a Sibelius paradox, it's a genome paradox exists. Look at this. Some amphibian genomes are larger than human. Some amphibians are not, but their genome number of genes are similar. So in the mammal, humans are here, okay? Some in the flowering plants, C. And some fish has more larger genome than human. Some even insect have a larger genome than human. But overall, when you compare the complexity here, the picked the representative species, as you can see, parasitic bacteria, single-cell eukaryote, multi-cell mold, worm, insect, bird, amphibian, mammal. So overall, genome size getting larger as the phenotypically complex, right? This is true. So organism with higher complexity tends to have larger genomes, okay? So this morphological complexity comes from adding new genes, okay? So from all genes, in eukaryotic genes, for essay, this is human gene, okay? Human 21% human genes can also found in prokaryote too, and single-cell eukaryote too. Well, that's interesting. What kind of genes that would be? DNA replication, translation, transcription, that really basic cellular function genes. Okay. Now, cell become complex. Okay. Now, new genes uptake for cellular compartment multicellularity. Okay. Now, development. So, there's more genes uptake. Now, vertebrate and animal-specific genes are added. So what genes are there? Immune system. Those genes are specific for vertebrates and animals. Okay. Now there's only vertebrate specific genes are added. It's a neuronal system. Okay. Neuronal systems are only found in vertebrate animals. So as you can see, increasing complexity is achieved by adding new genes for new functions. Okay. So here, single-cell eukaryotes, but multi-cell eukaryote add up extracellular gene function, function in extracellular, and also transmembrane function. Okay, this is basic. So gene function required for essential for every eukaryotic cell. Transcription, translation, metabolism, transport, and DNA replication, and protein folding, and cellular process, and others. Okay, but on top of that, new function, extracellular or transmembrane gene and proteins are there for higher eukaryotes. So human genome, when you compare human and chimpanzees, okay, 98.5% are identical. Really, really interesting, right? So 1.2% SNP, 1.5% inverse, and some chromosomal rearrangement. Okay, 25% proteins are exactly the same, it's just identical. Okay, and coding SNPs, coding sequence, there's less SNPs. Okay, you remember that. So exon evolves slower in negative selection pressure, right? Then relatively large change in gene structure and gene regulation, and 25% nucleotide substitution, and CG island. Using CG island, hotspot, right? You remember that, hotspot, CG island. And mitochondria is maternally inherited only, right? It only lines one. There's no recombination in mitochondrial genome, right? So people compare the human mitochondria to our relatives. Chimpanzees existing relatives. So 1,462 base pair difference between the human mitochondria to that of chimpanzees. Okay, those are non-existing human relatives. So human mitochondria is 386 base pair different from that of Denisovan, and 202 base pair different from Neanderthal. Okay, so there's some difference, but apparently Denisovan and Neanderthal looks like more closer to human than chimpanzee does, right? This is reference. Okay, people now found more, Denisovan and Neanderthal DNAs all around the world, and Eurasian there, continental. So this is how they sampled some. So yellow is Neanderthal, and this open square is Denisovan, the other. And they analyzed mitochondrial DNA. Mitochondrial DNA only. They found there is a common ancestor between Denisovan, Neanderthal, and modern human. Okay, then Denisovan may split earlier. Then at some point, the Neanderthal and modern human split just based on mitochondrial DNA. Okay, then they combine mitochondrial DNA and nucleic DNA, just gene, regular gene. Okay, and looks like the common ancestor again. Then is split. It's a common ancestor for Denisovan and Neanderthal. And they split here and modern human. And there's a genetic transfer between Neanderthal and modern human. Okay. So you remember a lot of several articles about the modern human genomes have a Neanderthal genome because actually they undergo sexual development each other, gets mixed together. So there's two hypotheses why there's no Neanderthal. Because modern humans are more adapted to the given environment. There's a Neanderthal faded away. Another things, so they hybrid, but eventually, so Neanderthal just faded away. But that's, I think, both in the hypothesis may be same. So Neanderthal would have been less adaptive. So it may have had less fitness compared to modern human. Okay, that's why Neanderthal became extinct, although modern human survived, even though there's a genetic information transfer each other. So if you use this to Google, there are a lot of interesting articles about human genome. You also find human genome evolution. So unfortunately, our closest living relative is a chimpanzee, okay? But still, although we seem together, it's really far. So again, unfortunately, we don't have really, really close relatives existing. Okay, now we look at the sequence, right? Sequence changes. Another change by evolution is gene duplication. Just another copy existing. Gene duplication occurs 1% of genome per million years. Okay, and after the nuclei duplications, another copy accumulates. Mutation, divergence accumulates 0.1% per million years. Okay, now silencing of one copy. One copy becomes pseudogene from active genes. Okay, this could take 4 million. So by looking at these duplication structures, you can predict how long it would take from here to here. Okay, so this is a globin gene complex in human. Alpha cluster, beta cluster, you remember that, right? In alpha clusters, there are 1, 2, 3, 4, 5, 6, 7 alpha genes exist. Where are they all from? Gene duplication, okay? 3 of which became pseudogene. Okay? Those only 4 remain active gene, but those are different. That's why if there's an identical, we should call alpha 1, alpha 1. But as the sequence are different, we call alpha 1, alpha 2, delta, theta, and gamma 2, right? And beta cluster, there are 6 beta genes there. Epsilon, gamma, G gamma, A gamma, tau, and delta beta. So one of them became pseudogene, okay? So there are 5 active genes in beta complex, okay? This is result of evolution, okay? What kind of evolution? Gene duplication, duplication, diversification, and pseudogene. Diversification and pseudogene, okay? So from like gamma globin to myoglobin to alpha beta globin and globin complexes, you see this kind of stuff, okay? Let's see. From like gamma globin, you remember like gamma globin has an extra exon compared to globin, right? From like gamma globin to globin, from plant globin to ancestral globin, insect globin, okay? Exon fusion occurred. And here now, gene duplication and diversification. Okay, now these two genes are unlinked. And another duplication, alpha cluster formation and beta cluster formation. This is evolution of a globin gene complex in eukaryotic system, from plant to insect to amphibian to birds to mammals. Okay. Again, let's move back. We know how long it would take for each event. Okay. And also how long it takes to accumulation of mutation in the gene. Then scientists calculate the year when this event happened. By doing that, scientists just hypothesize when amphibian evolved from ancestral insect, when mammal birds are evolved from other ancestral a common ancestor with reptile or other amphibians, things like that. So these numbers is not from random, okay? This is relative in precise based on precise calculation on DNA sequence change, okay? DNA sequence change may be precise, but okay, still this is kind of an assumption, okay? Prediction, okay? May not be true, but this is so far the best that you can measure. Pseudogen, some genes become pseudogen. How they become pseudogen? So processed pseudogen means they are translated, transcribed, and messenger RNA become cDNA, and cDNA is integrated there. That is one way, processed pseudogen. Another is non-processed pseudogen, just accumulation, mutation in promoter area or exon area, exon intron junction areas, whatever mutation occur, arrived there to abolish the function of the genes, okay? So now, there's another type of genome change. It's genome duplicate. Entire genome can be duplicated. It's polyploidization, okay? Ploidy. Ploidy. Haploid is N. We are human, a diploid organism. So we are E2N. That's called ploidy, okay? So genome duplication occurs to generate polyploidy. The only autopolyploidy means polyploidization resulting from mitotic or meiotic in the same species. Okay, become N, 2N, 4N, 8N, things like that. Allopolyploidy. This is polyploidization between closely related species, not the same species, but really closely related species. This is allopolyploidy. Okay, there's also aneuploidy, 2n-1, 2n+1, n+1, things like that. So human aneuploidy is disease actually called Down syndrome. Is one of the human aneuploidy, right? This polyploidy can be observed more frequently in plant than animal. So people use this polyploidy to generate diverse phenotype in crop. Okay, Cavendish banana is 3N. So wild type banana has very thick, rigid seeds. Those are not eatable. It's not good to eat them. But people generate 3N. 3N, this is odd number, right? Their meiosis is not correct. That means they can't make thick seed. That's why it's easy to eat for human. 3N, seedless watermelon, 3N, similar thing. There's no seed because of odd number ploidy. So strawberry is believed to 8N or 10N. Okay, wheat is 6N. Those are all, generated by human. In animal, so far, only salmon look like polyploidy. Salmon genome is 4N. Okay. In the early vertebrate, there's a 2R hypothesis. What is 2R? So early vertebrate genome underwent 2 rounds of duplication. 1 round of duplication, and again, a second round of duplication. Okay, so this genome, whole genome level duplication is also another driving force for evolution. So let's see what happened in why genome duplication is important. Here, first there's ancestral genomes. Okay, now genome is duplicated. Entire chromosome is duplicated, right? Now one genome starts losing genes. Okay, then some parts still remained, and this become diversified. Okay. And at some moment, this is integrated in the same chromosome or different chromosome. When you compare, so this is related sequence, right? It's here and here, right? Their sequence is very similar, not identical, because it accumulates diversification, right? So here, these sequences are related. In this case, this segment sequence is related to this segment, right? How you can find the gene duplication? You find a certain segmental DNA are conserved in the 2 different chromosomes in the same species, okay? So let's use the term that we learned last time, synteny. Array of genes, synteny, same synteny, can be found in 1 chromosome and the other, 2 different chromosomes, right? Why their syntenies are conserved? Because they are trace of gene duplications, okay? So there's ancestral gene duplication events. Eventually, there's a random loss occurs. Here, look at this. Random loss occurs, right? Now, this is the remaining part of the duplicated gene. Remaining part of the duplicated gene, okay? This is a single duplication event. If there's another duplication event, then there's a second peak here, right? Then start losing genes again. If there's one more round, then another peak there, right? Okay. In the vertebrate genome, there's 1 peak and second peak, that's it. How do you know? So this segment, you can find a certain segment existing as a 4 copies. See, 1 duplication give you 2 copies in certain segment of DNA, right? 1 more round may give you 4 copies of a certain segment. So this is the evidence that early vertebrate genome underwent the 2-round gene duplication hypothesis. It's a gene duplication, okay? This hypothesis can be, not really, can be supported by the existence of 4 copies of a certain segment of DNA, okay? So human genome, more than 50% human genomes are repetitive DNA, right? Transposable elements. So transposable elements increase in copy number, okay? But why they don't mess up the genes? Because big chunk of DNA comes in. Even more than 50% human genomes are transposable element. They don't have a gene. So actually they are checking negative selection. They are not really expressed because human cell has a really good system to suppress transposable element. Okay, transposable element names from transposition. This element comes out to move to another location, but those are completely silenced in the human genome by our own genome system. Okay, there is a separate chapter for transposable element. We'll cover that more detail. All right, this is the end of the lecture today.